This is a production of Cornell University Library.
Well, thank you for coming on such a nice sunny day. We're going to try to breeze through this in 5 minutes so we can all go outside, okay? I will try to get through this as quickly as possible. I realize that it's absolutely gorgeous outside. There's also some wonderful artwork upstairs that we can go to and see. Just a really neat collaboration that we did with researchers, art researchers, believe it or not, at the University of Wales on this topic of pollinator health. So there's some really great stuff upstairs, and I'll foreshadow some of that in this talk. Okay, so before I get started, I always make the wrong choice and put or I have in the past, put the acknowledgements at the end. So now I'm putting the acknowledgements at the beginning just to make sure that everyone who is doing all this work gets acknowledged. So to get us started here, I just want to give a big thanks to Aaron Iverson, who I don't believe is here because he's actually doing some of the field work right now. He's a postdoc in the lab. Nico Baert is our pesticide chemist. He sort of prides himself on not knowing much about bees, but a lot about chemistry. Ashley Fursch, who is an old lab manager. She's moved on to a position in the Carnegie Institute now. Maria Van Dyke, who I believe is here. Yeah, so I'm going to highlight some of Maria's extension materials. Paige Muniz, who is our current lab manager and may be in the room. She'll be coming later. I'm sure she's sleeping in. And then Emma Mullen, who is our senior honeybee extension associate here at Cornell. And we really have a wonderful team that I'm privileged to work with to show you a lot of the information that we're doing for research and also extension. Okay, so you guys are probably here because you at least have a physical connection to pollinators, and that you probably think that they're somewhat pretty, at least. I happen to think that they're beautiful. I'm going to show you two of my favorite pictures. So this is one picture I took of a mining bee, an Andrena species in blueberry when I was a postdoc. And then here's my other favorite picture that I've taken of a pollinator. So this is the American bumblebee, Bombus pennsylvanicus, that I took down in Arizona during the bee course down there. So they're really beautiful. You can also work with them. So here's some random researcher doing work with the honeybee, Apis mellifera, and they're wonderful pets to work with as well. Okay. And then later on, we're going to see how some people have taken this beauty of bees and brought it upstairs and actually made some artwork about those pollinators. So pollinators are very beautiful. They're also doing some very important things. So these pollinators, in addition to other insects that are out there, contribute a lot of ecological services or ecosystem services that provide benefits to us as humans. So that includes pollination, non-pollinators, these predators are doing pest control, and then also there's nutrient cycling that's going on in some of the detritivores out there. So this is estimated to, in the US, contribute about $57 billion per year to our economy. So these are not just beautiful insects, but they're also very important insects for the services they're providing. For pollinators, the whole reason that they are providing these services is because they're messy. Okay? So as a pollinator is going and foraging at plants, it's picking up a lot of this pollen and transferring it from a female plant to a male plant, or from the female part of the plant to the male, the male part to the female part. Let's get the basic biology down this morning. From the male part to the female part, or the female plant to the male plant to the female plant. And this is very important for general ecosystem services. So most of that green stuff that you see outside is an angiosperm, and a large portion of those angiosperms require insect pollination to be able to successfully reproduce. It's also worth a lot of money for agriculture, okay? So here in New York, these are our major pollination-dependent crops. We're called the Big Apple State for a reason. So apples are worth somewhere around 240, upwards of, the estimate for this year is around $300 million for this particular year, just annually. Soy, to some extent, is reliant on pollinators for reproduction. Beans, squash, cherries, strawberries, a lot of these very high-value crops that we should be eating more of in our diets are reliant on pollinators. So if you total all of that up and then consider the reliance on pollinators, so apple, for example, very reliant on pollinators to successfully produce a fruit. Soy, somewhat reliant on pollinators, but can also self. It can reproduce by not having an insect facilitate reproduction. If you total all of this up, we estimate that pollinators, and particularly bees, because they happen to be very good pollinators of these agricultural crops, contribute about $500 million annually to the state's production. Okay? Just to put this in some context for the world, so we estimate in the world that it's worth about pollination is worth about $170 billion per year to the economy, and somewhere around 3/4 of the world's crops rely to some extent on pollination. Okay? So that's, you've probably heard in the media that about 1 out of every 3 bites of food you take, a pollinator has been important to that. That's in large part because a large portion of the world's crops do rely to some extent on pollination. Okay? Okay, so in addition to the pollination services that bees provide, we also have honeybees that are contributing to our state's agricultural economy by producing honey. Okay? So we have in the state about 3,000 beekeepers, and they keep somewhere around 100,000 colonies of bees. On average, they're producing about 4 million pounds of honey each year, and that's worth about $10 million on average, okay? I say on average because this is the situation for the past 30 years in New York State. So this is New York State honey production on the y-axis here, and starting in, I believe, 1987, the trend in production all the way up until 2016, the last year that we have good data, okay? And what you can see here is that the last 13 straight years have actually been the worst honey-producing years in New York State's recorded history. So why is this occurring? It's part of a larger trend throughout the United States. So here are data from the Bee Informed Partnership. This is our major USDA-funded study looking at pollinator losses, or actually I should say honeybee losses in the United States. And what we've seen since taking data on sort of worldwide trends, but specifically United States trends, is that there's been somewhere between 1/4 and almost 1/2 of the honeybee colonies throughout the United States are lost every single year since 2006. This is a pretty massive amount of loss that is occurring. So is anyone a grower in here? Or related to a grower? Any crop, any crop. Yeah. So do you typically experience 1/4 to 1/2 of a loss of your Christmas trees on your farm every year?
Not every year. But
okay.
We sustain about 10% anyway. It's been a little higher in the last few years because of the weather.
Very interesting. And climate change is also linked to pollinator declines, which we'll talk about. Well, anyways, the point just to get across is that this is a very high loss rate for most agricultural systems, and bees are an agricultural animal. So this honeybee is a domestic food-producing animal classified by the USDA as a food-producing animal that should, hopefully not be lost at this rate to be sustainable. Okay? So how does New York do? On average, we're a little bit higher than the national average. So last year in New York, we lost 40% of our colonies. We've been up as high as 70% loss of our colonies. So of those 100,000 colonies that we have here in New York, we're losing, on average, about 50,000 of those every year since 2006. Okay? In addition to those managed honeybees, we also have a diverse fauna of wild bees here in New York. Okay? So here we have a list of all of the managed bees. So we have 3 managed species of bees, the western honeybee, Apis mellifera here. We also have the alfalfa leafcutter bee, Megachile rotundata, and a mason bee, Osmia cornifrons. All of those are managed. We have Bombus impatiens, the common eastern bumblebee, which is both managed and one of our most abundant wild pollinators. Those are the big bees that you see in the spring. Those big fuzzy bees is most likely the common eastern bumblebee, the queens that are emerging in the spring. And then we have 413 species of mostly solitary wild bees that we don't know that much about. Okay? But what we do know is 53 out of those 417 species, about 13%, are experiencing some form of population declines or range contractions, that is likely an underestimate simply because we don't have great historical records for a large portion of these solitary bees. Okay? So declines are occurring in both the managed honeybee and also these other bees, these wild bees. In addition, there are other pollinators that are experiencing declines right now. So here's a paper that was just published a couple months ago in Nature Communications. Here is the situation for bees in the United Kingdom, and you can see these declines that are occurring especially recently in bees. So this is occupancy, essentially a measure of their geographic range distribution in the United Kingdom. And you can see that hoverflies, if anything, are actually experiencing worse declines in the United Kingdom. In addition, it's not just pollinators. So this is something that's emerging in the literature right now, that we're noticing general insect declines. Okay? This is a paper, perhaps the most famous paper, that was published a couple years ago on this, showing more than 75% decline over the past 27 years in total flying insect biomass. This is in natural areas in Germany. This attracted a large amount of attention, so nearly 500,000 views in the first month this article was published. I'm lucky if I can get about 4 views in the first month that I publish articles. But this is now termed the windshield effect. So the idea here is that 30 years ago, you used to be able to drive around at dusk and have the privilege of getting a lot of insects splattered on your windshield. Nowadays, you drive around at dusk and you don't have as many insects splattered on your windshield. Okay? And we actually are working with DMV right now and Department of Transportation on seeing if we can actually get some data to say something about this here in New York. It's sort of an interesting collaboration going on. But this is something that is occurring. You know, you guys have probably heard about honeybee declines. Maybe you've heard about pollinator declines. In general, most people in the United States have not heard about general insect declines. This is becoming much more of a in the public view in Europe, but it's starting to emerge, and hopefully some of you in here have started to realize that this is becoming an issue in the US as well. If not, now you do. Okay, so it's all doom and gloom, right? Or hopefully it's not doom and gloom. What are the major stressors on pollinators? So why is this occurring? And if we know why this is occurring, what can we actually do about it? Okay, so we know that there are multiple stressors being faced by pollinators today. The first is pesticides. This is probably the thing you see in the news the most. Everyone wants to say it's the neonics that are killing the bees. Yeah, neonics are killing some bees, but it's not the only thing that's important. Okay, so I'm going to show you some data on insecticides, fungicides, and herbicides. We also know that pests and pathogens are very important as a driver of pollinator declines, especially novel pests and pathogens. So a novel pest and pathogen, for example, is this Varroa mite. This made a host switch from Apis cerana onto Apis mellifera, our western honeybee, sometime in the 1960s, arrived to the United States in 1982, and it turns out Apis mellifera does not have the co-evolved defense mechanisms against this parasite, and this is the major threat for honeybees in the United States and most of the world right now. Okay? We also know that climate change is important, things like phenological mismatch and droughts. So phenological mismatch meaning as climate change progresses phenology of the plants, some of those bees that are emerging at the same time and trying to target the particular plants are emerging at a later date. So the environmental cues that they're keying into are not the same cues that the plants are following, and therefore you're starting to have some mismatch. We have some good data sets supporting that in a few systems right now. We also know that habitat is extremely important. So if you have a low diversity or lack of resources out there, or nesting sites, especially for wild bees, you simply don't have the food or homes to support those bees. And then finally, for the managed bees, especially Apis mellifera, we know that poor management practices can lead to a whole host of problems. So I put this picture up here for a reason. Somewhere around 60% of all the honeybees in the United States get on 18-wheelers and make the trip across to California every single February. Okay, they go pollinate almond, so we can all have our wonderful almonds to eat in our cereals or other things. Almond is 100% reliant on honeybees for pollination. There's a few other managed species of bees that are starting to make some inroads in the industry right now, but in large part, it's mostly just honeybees. That's 60% of the colonies in the United States are getting on 18-wheelers in February and making that trip out to almond. Well, it turns out if you're a tiny little insect on an 18-wheeler making a 4-day trip out to California, it is a little bit stressful. Okay, so the emerging theme here in the research is that there are multiple stressors that are important for all of these problems associated with pollinator health. And also there's interactions. And the interactions seem to be very important as well. So just as an example, let's say that western honeybee took a trip out to California. It gets out to California. This isn't actually California, but just make believe that's California. So it's in almond. It has a low diversity floral resource, okay? So it's basically just eating almond while it's out there. We know almond is pretty stressful on bees. So the pollen has a lot of amygdalin in it, some secondary compounds, and it's not very nutritious. At the same time, it's exposed to pesticides because growers are treating their plants with fungicides during pollination, and the bees are exposed to those fungicides. They're also exposed to insecticides as the residues in the pollen and nectar from things that were sprayed even the year before. In addition, those pesticides, we know, cause the bees to become more susceptible to pests and pathogens. And by the way, while you're out in California, you've got 60% of all the honeybee colonies in the United States out there, and you're foraging in an area where there are colonies that are very sick compared to yours, and the pathogens are being transmitted at those flowers. So you have this complex interaction that's occurring oftentimes for bees. Okay? All right, so any questions before we start? So what I'm going to do is I'll spend the rest of the talk going through 4 of these particular stressors, some of the things that we're doing for research, and then trying to pass along some information for particular things that you can actually do. Okay, so what are things that you can do to improve habitat? If you're a beekeeper, what are some management practices that you can do? If you're a grower or you do use pesticides, what are things that you can do to minimize risk to bees? And then I'll finish up with a little sort of vignette on spillover of pathogens between honeybees and wild bees and some things that we can potentially do to stop some of that. But before I get started, any questions? Yeah?
Are wild bees not in colonies? They're just by themselves?
So I could give you a whole lecture on all of the different life histories of wild bees, but the short answer is that yes, most wild bees are solitary. And Brian Danforth, who is in the back here, spends most of his career discussing solitary bees. So if you want the 45-minute lecture after this on solitary bees, then yes, talk to Brian. But yes, we have a few species. I don't actually know off the top of my head exactly how many in the state are social bees, but it's somewhere on the order of about 50 or 60 social bees in New York.
That sounds about right.
Yeah.
Genus dialectus plus bumblebees, yeah, about 60.
Yeah, so right around 60 social bees. And then those other species are solitary, and that's true for the world as well. So the vast majority of bee diversity is solitary bees that are not in a colony like you typically think about for honeybees. Yeah?
Does that classify mason bees? Are they, because I know that they go into tubes.
Yep. And if you're around tomorrow morning, you're going to actually be able to make a mason bee tube nest that you can bring home. Okay, great. If anyone else wants to, you as well.
But I was curious because they do colonize because more than one bee will be in those tubes.
Mm-hmm. So there can be aggregations of mason bees, but that is a solitary bee. So there is one female who is filling up that tube with her offspring. Yep. Okay, let's take maybe 2 more questions and then get on just so we don't go too far over. Yeah.
Where do the bees that pollinate the almonds come from, 18-wheelers?
They come from all around the country. So the vast majority of commercial beekeepers in the country who are distributed all around the country will make that trip, and it's all due to money. So they get a lot of money. So each colony that goes into pollinating almond, they typically get somewhere around $180 right now. So that's $180 per colony. It's about 1.6 million colonies that are going to pollinate almond. You can do the math and see why they're going there. All right, maybe one more question. Yeah.
You mentioned that bumblebees are sometimes managed. Was that correct? Did I hear that correct? How or what? I mean.
Yeah, so the major reason that bumblebees are managed is they do something different from honeybees. They can buzz pollinate. So does anyone have a Sonicare toothbrush? Yeah? That is an ultrasound frequency that toothbrush is doing to remove the plaque from your teeth. Bumblebees can do that as well, not in your mouth, hopefully, removing plaque, but what they can do is they can go up to solanaceous plants that have poricidal anthers, and those anthers require sonication, so ultrasound frequency, to release the pollen. Those bumblebees can do it, honeybees cannot do it. So all of those tomatoes that you guys buy in the grocery store, the vast majority of those are buzz pollinated by managed bumblebees in greenhouses around the world. Okay? All right, let's move on, and then we can do some questions at the end too. Okay, so we'll start with habitat. All right, so what do we know about habitat here in New York or elsewhere, and then what are some things that we can potentially do to improve habitat? Okay, so first we'll start with what do we actually know about habitat. So I'll show you some data from a master's student in my lab. So this is Nelson Milano. And his major question was, okay, what habitats, just stereotypical habitats here in New York, where do bees thrive and where do bees not do super well? So what he did is he put out 64 colonies into 16 different sites across this region of New York. So here's Ithaca right here, Syracuse, Geneva, and Rochester. So replicated in each geographic region here, he put colonies into conventional agricultural areas, organic agricultural areas, natural areas, and I realize that's a broad definition of what could be natural, but we tried to get it sort of your stereotypical natural area here. And then also urban and suburban areas, okay? And he did this across 2 years, so there's actually 128 colonies over 2 years that he put these bumblebees in there, and just saw how they performed. Here's a picture of how the colonies looked when they were out there. And what he found was a really striking result, okay? So across all of these different types of habitats that these bees were in, this was suburban, organic ag, natural, and conventional, he found that in suburban areas, the bees don't do very well. So they don't grow and they don't gain weight, and therefore their fitness is very low in the suburban areas. In contrast, these organic agricultural, natural, and conventional areas all do about the same. Okay? So we were wondering why that might be. Why is it that the bees might not be doing very, very well in suburban areas? And I have a very talented postdoc in the lab, Aaron Iverson. He's so talented that he's actually out there doing work right now while we're in here talking about his work. But what he's done over the last 2 years is he's developed a land cover map of floral resources for this region of upstate New York. So he's gone and done surveys of 462 plant species across 20 different land cover types, which are shaded in different colors here, to then come up and integrate how many flowers are available at different points of the season. So this is what those data look like for the 20 different land cover types. So here's hay, perennial crops, conifer forest, row crops, lawns, roadside ditches, etc. And you can see these floral resource curves that really sort of map what, intuitively you might think might actually occur out there. So especially early in the season, we have these tree resources, which lead to a huge spike in floral resources. Midsummer, we have a lot of sort of roadside ditches and disturbed areas that are contributing floral resources. And then in the fall, we have the old fields kicking in with a lot of these asters and solidagos that are leading to a lot of bloom. So we looked at a lot of different things in addition to these floral resources as what might be predicting the performance of these bumblebee colonies. And I can show you the results with a picture as opposed to a graph. This is the reason why colonies likely are not performing very well in suburban areas. Okay? We have a lot of suburban areas where we think these green lawns are beautiful. I guess the question is, why don't we think that green and white and pink lawns could be beautiful? There's a lot of turf in these suburban areas, and there's just not a lot of resources for bees in these places where most of us actually live. Okay, so what can we actually do about this? Well, it turns out there are some people already doing something about this. So you could go out, and I've been advocating for a while now, just don't mow your lawn as much, but I realize some of people have this infatuation with wasting their lives mowing their lawn. Some relatives are in this room right now, and I'm saying that for a reason. But so there are also extension programs, for example, in Minnesota, that are promoting bee-friendly lawns. So they're promoting the inclusion of some of these trifolium species and various other legumes that are very good resources for bees that can still be mowed and regrow fairly well. So just as an example, this actually just came out a couple days ago, so about a week ago now. Minnesota just committed almost $1 million. This is a state-funded program to pay for homeowners to transform their lawns into bee gardens and species as the species inches closer to extinction. So they have one particular bumblebee species that they're very concerned about. Okay? So that's something that you can potentially do. You can promote your lawn or mow it a little bit less. In addition, we'll talk a whole bunch about this later, but this is one of the pamphlets that you guys have here. If you need your lawn, you need to play soccer on it, whatever it is that you need your green space for, you can also create a pollinator-friendly space, okay? So this is something that would be set aside from your lawn. The two key points, so we have a one-pager that you guys can read about some of our recommendations, and there's lots of resources out there as well. But the two key take-homes are to choose a diverse array of pollinator-friendly plants, okay? So pollinators love diversity. It's not that, if you have something that blooms for 2 weeks in July and that's a really good plant, well, that's the only thing you should do. What you should do is try to plant something that blooms all the time. Try to plant lots of different species of plants that provide a lot of nectar and pollen for those pollinators. And then the second thing that most people don't really consider, but remember, we have lots of bees, and lots of them are solitary bees. Many of them are ground nesting bees, or some are stem nesting bees, or they make their homes in various different things. So try to create a habitat where wild pollinators can actually nest as well. And we have some recommendations in here for how you might be able to do that. Okay? All right. And then one thing that was touched upon just a little bit earlier is we're making an effort right now to try and understand if we're going to be putting large tracts of land aside for potentially improving habitat, we're trying to understand which seed mixes really are the best and what benefits, how are those actually benefiting wild bee populations. So one example of this is the solar projects that are starting to increase here in New York. Governor Cuomo has a goal of 6,000 megawatts, or about 40,000 acres of new construction in New York by 2025. So we're working with several solar companies and a few other land management people to try and understand, well, if you put these pollinator-friendly mixes down here, which are the best mixes for bees? Okay? All right, so that's habitat. Next, what we'll do is we'll go to the most controversial topic, pesticides. And this is what I spend a lot of my time talking to growers and the public and regulatory agencies about lately. Okay. So we'll start off with something that beekeepers, when I first started this position 4 years ago, beekeepers came to me and said, Scott, when I go in to pollinate apple in New York, I come out and my bees look horrible and it's the pesticides killing the bees. And I said, okay, that could be the case. It could be that the pesticides are killing your bees, or it could be lots of other things. It could be that they really don't like apple pollen. It could be that they're exposed to all sorts of pathogens when they're in apple. It could be lots of things. So what we did is we said, well, listen, why don't we try to address this topic? Let's actually quantify what pesticide risk is to bees when they go in and do apple pollination. So when those trucks show up at lots of different apple orchards around in New York, what kind of pesticides are they actually exposed to? So what we did is here's a little cartoon showing what we did. So we put 120 experimental honeybee hives into 30 different apple orchards. So there's four hives per orchard. It was basically just a slide showing you that this was a lot of work. And some people in the back of the room have been doing some of this work this year for a follow-up project. Thank you very much to the back of the room. So what did we find? What I'm showing you here is just sort of a one-slide summary, because I want to be able to discuss other things as well. But what I'm showing you here on the y-axis is a pollen hazard quotient as a percentage of a contact LD50. So what does that mean? Basically, what this is saying is we're taking all the different pesticides that we found in the pollen that those bees were collecting, and we're summarizing it not just in terms of how much of those pesticides were there, but also how toxic each of those pesticides were. So for example, imidacloprid, a neonicotinoid insecticide, is very toxic to bees. Just a tiny amount of imidacloprid is very toxic to bees and will contribute greatly to a percentage of their contact LD50, when 50% of those organisms, or these bees in this case, die acutely. Whereas you can be exposed to a whole bunch of glyphosate. Okay, so glyphosate being Roundup, it's an herbicide. You can be exposed to a whole bunch of that, but it's just really not very toxic to bees, so therefore it doesn't contribute much to risk, which is this hazard quotient. Okay? So what we can look at is this pollen hazard quotient. So when we go and sample the colonies that were out at these orchards, what is in the pollen? What are these bees actually being exposed to, and how risky is it? What we can see is a bit of a mixed result. Okay? So generally, we have relatively high risk from pesticides at some of these orchards. So 80% of a contact LD50, that means about 40% of the bees in that colony are dying within 24 hours of being exposed to these pesticides at that particular orchard. Whereas if you're down towards 0, or about 0 up to about 3%, there's a pretty low risk for acute exposure. Okay? So what we see is 2 of our orchards are above a US EPA acute exposure level of concern. 5 of those orchards above the European Food Safety Authority acute exposure level of concern. And 22 out of the 30 orchards are above this European Food Safety Authority chronic exposure 10-day level of concern. We had these bees out there for 10 days, which is a normal pollination window, so that's an apt estimate to be putting on these. So what we can see is some orchards are doing fine, but somewhere around 2/3 of the orchards are above some regulatory agency level of concern for pesticides during apple pollination here in New York. Okay? Okay, so that's during pollination, this potentially very risky time that bees are going into these orchards, providing services so we can all eat our apples and other very nice things if it's fruits or vegetables. What about just normal pesticide exposure to bees? So what we did is we formed, as part of the New York State Pollinator Protection Plan back a few years ago, we formed something called the New York State Beekeeper Tech Team. And you can go to this website if you're interested in more information on this tech team. We do a lot of things, and I'll highlight one more thing that we do with this tech team. But one of the things that we do is we assess pesticide exposure to bees. Okay? And the tech team is comprised of Emma Mullen, our senior honeybee extension associate, Paul Cappy, our New York State apiculturist, Travis Grout, who's our economic analyst on the tech team. So he's actually providing not just the biology expertise, but also the economic expertise. How does a beekeeper make decisions about what to sell and what to buy? And then myself as the researcher. Okay, so what did we do? We went out and sampled 198 beekeepers this first year that we started the tech team, and we did the same thing that we did with the apple study. We went out and we sampled, this time, the wax to see what kind of pesticide residues are typically in wax. Here's Nico in our pesticide detection facility. We quantify somewhere around 270 compounds right now. So basically anything that a grower sprays here in New York, we can know what that level is in whatever matrix we're looking at, in this case wax. So are bees typically exposed to pesticides? Yep. So on average, we find about 5.5 pesticides per colony in New York. So here's a histogram showing the number of pesticides across hobbyist, sideliner, and commercial beekeepers. So hobbyist, a few colonies, sideliners, up to about 50 colonies, and then commercial beekeepers, you're really making a business out of this. This is your full-time job, and you have a lot of colonies out there. So pesticides are ubiquitous. We found at least one compound in 197 out of 198 colonies. So there's one hobbyist out of 198 samples that we did not find pesticides in. Okay? What about risk? So now what I'm doing is I'm putting this hazard quotient on the y-axis. So this is a percentage of your contact, LD50 again. And what we can see here is a very different story. So now it's not apple pollination where it's, some of these samples are upwards of 80% of a contact LD50. Now we're down generally below 1%. So these are individual colonies from each beekeeper here. generally fairly low risk to bees. Okay, so pesticides are there, just like they're there in the fruits and vegetables that we eat, but they're very low risk. But what we know regarding this hazard quotient is it makes the assumption that everything is additive. So there's no interactions among pesticides that are actually occurring and contributing to risk. And we know that's a bad assumption. So number one, pipernyl butoxide was found in 99% of our samples. That actually is a pesticide synergist. So growers will put pipernyl butoxide in a tank mix to make the insecticides more toxic for whatever pest that they're trying to control. In addition, we see a lot of fungicides. So what's here in purple as our sort of 13 most prevalent compounds here, we see a lot of these fungicides that are known to be synergists and cause insecticides to become more toxic. In fact, 82% of the colonies that we screened contained a pesticide combination known to be synergistic, and that's just what we know is synergistic. So what does a synergism actually look like? I'm going to highlight Aaron Iverson again. So Aaron doesn't just do floral resource curves, he also is a laboratory biologist. So here are some survival data from a bumblebee feeding assay that we performed. So what we did is we exposed bumblebees to either a control, so just a sucrose solution, and looked at their mortality over time. So this is hours on the x-axis and then probability of survival here. And what you can see here is in green is that, yes, bumblebees survive great when they consume sucrose. There's very little mortality. And same thing with fungicides. So in blue here, you can see that the bees do not, perish from eating fungicides. And that's good because bees aren't fungi. Insecticides, so this bifenthrin, a pyrethroid insecticide, there's a little bit of mortality. So now a little over 25% of those bees are dying acutely in this particular trial. But what happens if you mix that fungicide, myclobutanil, with bifenthrin? Now all of a sudden you have greatly increased mortality. Okay? This is what that synergism is showing. This is a fungicide causing the insecticide to become more toxic. We think this is likely the reason why we're finding some of these landscape patterns for fungicides. So myself and a few others, including Christine, who's in this room right here, did a large-scale analysis on bumblebee declines in the United States a couple years ago. So especially focusing on these major bumblebees right here, the rusty patch bumblebee, Bombus accidentalis, Pennsylvanicus, and Terricola. And what we found is the best predictor of losses of those bumblebees, so losses shown in red here from their historical range, and then whether they're present shown in black here. The best predictor of losses is what's shown in blue, and that's fungicide usage across the United States. Here's the figure showing this. So if you spray more fungicides, you are greatly increased probability of loss of those particular bees. Okay? This is largely driven by one dominant compound, chlorothalonil. We know a little bit about chlorothalonil, so we know chlorothalonil causes fluvalinate to become 5 times more toxic to bees. So here's your expected mortality with chlorothalonil and fluvalinate, so mortality at day 4. But here's the actual observed mortality. So there's this synergism occurring between fluvalinate, a pyrethroid, and chlorothalonil. How much chlorothalonil is used across the United States? A lot. It's the most used fungicide in the United States. Okay? So this is the amount that was used in 2009, which is relevant to our data. A little bit, almost twice as much as the next most used fungicide in the United States. Here are the 3 common trade names, Bravo, Echo, and Daconil. We also know that this is true today. Okay, so there's a little vignette on some pesticides. The question is, what can we actually do about this? So I think the first thing that we can do is we can increase awareness of this issue. So I spend a lot of my time talking to growers and talking to various people, pesticide applicators, people who use pesticides, trying to just get this information out there. Okay, this and many other vignettes that we have. One sort of neat way that I think this is a new way that we've been trying to communicate this is something that's upstairs right now. So this is a collaboration that we had with Annette Townsend. So she's one of these artists from Wales. And what we did is we sent wax from one of these studies from a honeybee colony placed next to an apple orchard during bloom to Annette and said, what would you like to make out of this if we gave you some of this wax? And by the way, we quantified pesticide residues in the wax, so we can tell you exactly what's in there. This is what she came up with, which is upstairs right now. So this is the title of the piece. It's called Wild Strawberry for Fragaria vesca. This is what it looks like. And here's what's right underneath the title. Beeswax, pesticide residues in the beeswax, piperonyl butoxide, fenpyroxamate, metolachlor, azoxystrobin, coumaphos, pyrostrobin, cyperdinyl, trifloxystrobin, fluopyrim, and atrazine, tinned copper wire, tissue paper, dressmaking beads, cotton threads, silk fabric, dry ground artist pigments, acrylic paints, and acrylic varnish. And then starred are these pesticide residues were also found on nearby strawberry flowers. I think this is sort of just a neat way of conveying a message that's not typical for scientists. So this is, a really neat science-art collaboration. So she posted this on her Instagram page, which I'm too old to understand why that's important, but it is important nowadays that you have an Instagram page. And it was picked up by the BBC. So they said, boy, that's really neat. Could we come out to your studio and highlight this a bit? So she was just on Countryfile, I believe a week or two ago. And this is a program that's viewed by about 5 million people in the UK, each program that they have. And this is what she did. So she showed this, boy, I should know the name of this person because I think she's fairly famous. But this is the person who runs Countryfile. And she showed her how she had actually made some of these things. And this whole project was highlighted on this program called Countryfile. Okay. So we can increase awareness of this issue of pesticides, maybe via some sort of unconventional ways with artists. We can also increase knowledge. So Maria is sitting right here. This is a booklet that she has spent a lot of time on over the last year, and it's an incredibly useful resource for growers. So what she has done is she has done this for tree fruits and landscapes and ornamentals, and also small fruits now, almost small fruits. Hey, all right, good. But what this booklet is it's a systematic literature review of every single thing that a grower can potentially legally spray on their crops. And then the review has looked in to say, what do we know about synergisms between fungicides and insecticides? Let's highlight that for growers so they know if they're potentially tank mixing a fungicide and insecticide, which is common, which ones pose the least risk to bees. Okay? This is a really impressive resource, and you can download it here. So this is what I spend a lot of my time doing now, is going to growers and telling them, here, take these booklets, tell your friends about them. Here's a way that you can make more informed decisions of how to target your pests without harming pollinators or posing minimal risk to pollinators. Okay, and then finally, so we can try and get people more aware of the issue, we can provide them with resources, and then regulatory agencies can also step in. So this is sort of a final thing that could potentially occur to ban the use of particular pesticides that might be harmful to various organisms, including bees. So I was very surprised to see that just about a month ago, so in late April of this year, the European Union has followed up their bans on neonicotinoid insecticides. And what they've done is they've taken some of our research that was highlighted here. So this is an article that was just in The Guardian. But they've taken some of our research and other research, and they've banned chlorothalonil in Europe now. Okay? This is the first time I've ever seen a fungicide banned for its potential non-targets impacts on animals, which I think is very interesting and potentially very controversial. This is the most used fungicide in the United Kingdom and most of Europe. It's also the most used fungicide in the United States. So I'm interested to see what happens, if anything, here in the US, which is typically quite behind Europe in terms of environmental protections. Yep.
Chlorothalonil first started to get heavy use, what year?
When did it start to receive? It's been used for quite a while. I'm not sure, I can tell you that for neonicotinoids. I'm not sure when that really started to take place for chlorothalonil. But I can get back to you on that. Yeah.
What are the potential substitutions for chlorothalonil?
That's a great question. That is a great question. We know a little bit about that here in New York because we are doing a risk assessment for neonicotinoid insecticides and some other pesticides, and we're trying to come up with what those substitutions could be. Typically, what's happened in Europe and with neonics and also here is they've made these restrictions without thinking about what the substitutions might be and then the risks that might be posed from those substitutions. I think common sense would be growers are not just going to stop using pesticides. The vast majority will. They will substitute something else. And if we don't know what they're going to substitute to, well, maybe we should consider that before we ban something. Europe has been not really doing that. They've been sort of just banning things and then seeing what happens. We're trying to do that, and Canada has been trying to do that as well. Yeah.
This is mostly commercial that you're talking about, because I don't know Bravo or Echo or Daconil. Mm-hmm. Are there products that contain this that are used more in home gardening? Sure,
those are used in home gardens. You can buy it at Lowe's, Home Depot right now. I have one, I actually used it. I shouldn't tell you this, but I have used it. It's a very effective broad spectrum fungicide. And you can buy it in your box stores, but you're absolutely right. It's mostly used, as most pesticides are used, it's mostly used for commercial production.
Neem is also a fungicide, do you know if that's one of the?
Yep, some neem oil is a natural product. It's not a synthetic pesticide, so it is approved for organic use. There are almost 1,000 different pesticides that are used, and that's just active ingredients. There's also about 5 trade names for each of those that are used in the United States. We could talk for a long time about various products, but if you want more of those resources, go check this out. Okay, so that's pesticides. Oh boy. And then what I'll do is I'll finish up here with management practices and pests and pathogens, and then we'll go upstairs, we can check out some art, and then hopefully we can go outside and spend the majority of the afternoon in this beautiful sunny day that we're having here. Okay, so a quick vignette on management practices. So let's get back to the New York State Beekeeper Tech Team. So here in New York, we have this great state-funded program where we've been working with beekeepers since 2016. Here's just some pictures of us going out. This is Emma Mullen right there, and this is Travis Grout, our agricultural economist, who we've been making work with bees lately. And then a beekeeper, John Ryan, right here. And I can't actually tell who those are right there. So anyways, the major thing that we found, in addition to pesticides and some other things, is the Varroa mite. So the Varroa mite on honeybees is clearly at epidemic levels in New York State and elsewhere. This is the major problem for honeybees right now. We don't know what the relative importance of of stressors on most other bees are right now, but the Varroa mite for the Western honeybee is the problem right now. Okay? So we can see, this was our first data that we took in 2016 when we started the tech team. We can see among hobby beekeepers, sideliner, and commercial beekeepers that they are almost always above the threshold of 3 mites per 100 bees. If you have a 3% infestation with Varroa mites in the fall, you're likely to lose that colony to the viruses that get passed on from that mite into the bees, and your colony will likely fail that winter. Okay? And it's also true here in northern New York, central New York, and western New York. Here are some data just showing, again, of all of the different factors that are associated with survivorship of the colonies, now that we have 2 years of data, full years of data, showing what is predictive of colony losses. We can show that if you have higher Varroa mite levels, your probability of winter survival goes down. Okay, so Varroa is our best predictor of overwinter survival. That's not just on a national level data. These are our own data here in New York from the New York State Beekeeper Tech Team. And again, it's largely because those mites are vectoring viruses such as deformed wing virus. You can imagine why a bee is not doing very well if it looks like that. Okay? We also know that beekeepers who treated their colonies for Varroa had lower Varroa levels. This is not rocket science. So if you have a Varroa problem and we have tools for you to treat for Varroa out there, you can reduce your Varroa levels. So if you treat more often, you have lower Varroa levels and you potentially have greater survival of your colonies. What we've seen now, so this is about a third of honeybees in New York right now. This is about 28,000 colonies, so somewhere between a quarter and a third of the colonies that are wrapped up in the New York State Beekeeper Tech Team as part of New York. Here are the loss rates in the winter loss rates, summer loss rates, and annual loss rates in 2017. We've been working very intensively with these beekeepers to try and get them to improve their Varroa management practices. I'm not showing you all the intermediate slides in here to show that, yes, they have been monitoring more, they've been treating more, but what we can see is that we've got about, about 11% decrease in overwintering mortality. This is a large number of colonies that are now, of course, this is observational data, so we can't prove that this is actually the case. It could be year-to-year variation, but there is a large number of colonies that improved their health last year, potentially due to these management practices. We think this intense state-funded program appears to be benefiting beekeepers and especially their bees. Okay? So if you are a beekeeper out there, treat for Varroa. Okay, and then I'll finish up. And then we can go upstairs. Well, I can take some questions and then we can go upstairs. But I'll finish up on showing you some connections between management practices and also pests and pathogens, and why it's not just a beekeeper problem, this Varroa mite, but it's actually a total pollinator health problem in the broader community. Okay, so there's two questions that we're going to address. So how do bees actually transmit disease? And then can poor beekeeper management practices influence overall wild pollinator health? So you would think we would know more about how lots of different things transmit disease. We even know very little about a lot of human diseases and how they're transmitted. Okay? For bees, we know almost nothing. But the theory is that, let's say you have a bee out there, it has some blue pathogen particles either on or inside of it. It goes and visits a flower. It potentially transmits those pathogen, or deposits those pathogen particles onto the flower. Then another bee comes on, potentially from a different species, picks up those pathogen particles, goes on its way, and potentially becomes infected. Okay? So we wanted to know, there's some laboratory evidence and field cage trials that might actually be taking place. But we wanted to know, well, how many parasites are actually out there in bees, in lots of different species of bees, and also on flowers in nature? Okay? So we said, what's the prevalence of parasites in these plant pollinator networks? We went out to 3 old fields here in New York, and we sampled a lot of bees and a lot of flowers. So almost 3,000 individual bees from 110 different species, and almost 3,000 flowers from 92 species. And then we screened them for 5 parasites. So this was a project funded by NIH, and we still have a whole team of people that are working on this project, even in this room right now. Here is a heat map showing the data for bees. So this is our 120 species of bees broken down by genera right here, and then the numbers are just how many individuals per genera, per genus that we screened. And what you can see here, this is prevalence of each of these 5 parasites. And the basic take home here, I just wanted to show you that there's a lot of parasites in a lot of different genera and species of bees. Okay? So 42% of bee species have one of these 5 parasites, at least one of the 5 parasites, and 16% of individual bees have a parasite. Okay, what about flowers? If we screen flowers, do we actually see these bee parasites on flowers? And this is perhaps one of the most interesting pieces of data I've seen, I think, in the last 5 years or so. So yes, so same situation here. We're looking at the different genera of plants here. There's 120 species that are encapsulated here, and 5 different bee parasites with colors according to the prevalence. And what we can see is 70% of flower species have a bee parasite on them, and 14% of individual flowers have parasites. About 1 in 6 of every flower that you will see, that you come along during this pollinator walk, is likely to have a bee parasite on it. That's a lot of bee parasites. That's like sort of a fundamental different way of looking at flowers, I think. These are not just these beautiful, resources. They're also potential transmission venues for various parasites that the bees might get. Okay. So I'll finish up with this message, which I give to a lot of beekeeping organizations. There's sort of this movement in the beekeeping world right now regarding Varroa. It says, You either treat a lot for Varroa and you control for Varroa, or if you're in the different camp, you let Varroa kill your weak colonies and have natural selection take place. So then you keep the strong colonies, but you let the weak colonies die. In the process of letting those weak colonies die, you have these disease bombs that go off, okay? So my response to those people is this. What we can do here is we can look at all of the different bees. So this is a plant pollinator network, including all of those 120 species of flowers, 120 species of bees and 92 species of flowers. And we've looked at the interactions, okay? So these are all the people that are working on this. We have Chris Myers here in the physics department, Pete Greystock, a postdoc who is now in the UK, Steve Eldner, a theoretical ecologist in ecology and evolutionary biology, and Paige Muniz, our bee taxonomist and lab manager. So what does this, just again, what is this figure actually showing? So these are all, at each of these nodes right here, these are all the different bee species that are interacting. These are all the different flower species over here, okay? And then if you have a line drawn between them, that means this particular bee species interacted with that particular flower. And you can see there's a lot of crossover. So, for example, Apis mellifera interacts with a lot of different flower species, and then there are a lot of those other species of bees that interact indirectly via Apis mellifera via the plants with Apis mellifera, okay? So what if you let your colony fail from Varroa? It gets deformed wing virus, for example. You potentially have a very sick bee that's going out and going and visiting all of these different flower species. Here's what happens if you look at how all of these indirect interactions. So this is the bounce back into all of the different species, now highlighted in green, who interact indirectly with Apis mellifera. And what I've layered on top here, these are the flower species with quote-unquote honeybee parasites, so parasites that were in the honeybee that we screened for. And in red here are all the wild bee species who share those parasites with the honeybee, okay? What we saw overall is that about 63% of flowers visited by honeybees have honeybee parasites on them. 78% of wild bees that visit those same flowers have honeybee parasites. We potentially have a large problem if you are letting your colonies just die from Varroa. They are not just influencing your operation, they are potentially spilling over and causing a pathogen spillover threat to the entire wild pollinator community. Okay? This is the message that gets hobby beekeepers to take care of or actively cull the colonies that are being ridden by Varroa. All right? Okay, so I'll finish with that. You guys have been very tolerant of me going 5 minutes long. But I'll just point to a couple more resources before we end here. So here is our main extension webpage, so the Cornell Pollinator webpage, pollinator.cals.cornell.edu. You can also become our friend on Facebook if you would like to. We have about 1,500 friends right now. You can become our 1,501st if you're on Facebook and would like. We update the Facebook page 2 to 3 times a week with some sort of new bit of information. So you can just receive information passively if you'd like. And then just thanks again to everyone in my lab and some of the funding agencies who have supported this work. Thanks very much for your time.
This has been a production of Cornell University Library.